chemistry of pesticides, microsomal monooxygenase systems, extramicrosomal metabolism of insecticides, insect resistance to insecticides, pesticide residues. For all these 5 subtopics under metabolism, resistance, and residues, okay, I have given you the overview. Uh, but today we are going to talk about synthetic insect control agents. Under synthetic insect control agents, we have covered in our last class, previous classes, we have covered about these things where we have studied about a few important things there. We have studied about different aspects like alternating agents, chemosterilants. Under chemosterilants, we have studied about alternating agents, non-alternating agents. And under non-alternating agents, we have covered phosphoramides, melamides, and all these things. Then after we have studied about insect growth regulators, where we have covered chitin synthesis inhibitors. Under that, we have studied some of the examples like benzoyl phenyl ureas. Can you able to recollect? We have studied different things under insect control agents. Then after we have studied about GNL hormones, followed by decent agonists. Okay, all these things we have studied in our previous class. Okay.
Yes, madam.
Okay. And we are under this particular unit, synthetic insect control agents, we are left with 4 topics. The first one is attractants, second one is sex pheromones, and the third one is insect repellents, and the fourth one is antiferents. We are left with these 4 topics under synthetic insect control agents. Okay. As a revision, okay, I want to tell you what all we discussed within 5 minutes under the first aspect of synthetic insect control agents, that is alternating agents. In alternating agents, we have studied about Affinitepa and Metepa, which are the valuable tools in the arsenal of insect pest management, where by using sterility in target insect population, these alternating agents offer an environmentally friendly alternative to conventional insecticides. And their use in sterile insect technique programs demonstrate their potential to sustainably control pest populations. Although careful application and monitoring are essential, to maximize their effectiveness and minimize resistant development. And further, we even have studied about 2 examples, that is hempa and thiohempa, which are the significant phosphoramides under non-alkylating agents, which are also used as chemosterilants in order to control insect population by increasing sterility. And then after we have studied some of the important things under non-alkylating chemosterilants, examples aminopterin, fluorouracil, nitrogen mustard, etc. And not only that, we even try to cover melamides, dithiobutates, dithiazonium salts, and other miscellaneous compounds. And then after we have studied about insect growth regulators, where we have studied how the chitin synthesis occurs, how the chitin is acting as an exoskeleton in the insects. And under this chitin synthesis inhibitors, we have studied about benzyl phenylureas, which are very active and effective insect growth regulators, which disrupt the insect synthesis of chitin, a vital component of the insect exoskeleton. And not only that, we have studied about bofrofacin, which is also used to control the certain insects by targeting the chitin synthesis. And then after we have studied about GNL hormone units, Under this, we have studied two examples, methoprene and phenoxyetrar, which are playing a major important role in disrupting the insect growth and development. And apart from that, we have studied about edison agonists. The example for the edison agonist is tebuconazide, okay, which is particularly controlling lepidopteran pests in agriculture. This is what we have studied in our previous classes. Coming to the today's class, we are going to talk about the first one is attractants, as you are seeing in the screen. These attractants are the chemical substances that are used to attract animals or insects or other organisms. They can be natural or synthetic, which are commonly used in agriculture, pest control, and even in research. There are different types of attractants which you are seeing in the screen. The first one is food-based attractants, and the second one is pheromones. Actually, here we are talking about pheromones, but specifically we are going to talk about the importance of pheromones after the completion of attractants in our topic in our today's class. Okay, but the attractants, under the types of attractants, we are going to study about pheromones also, but in a very limited way, I mean, in a short way. Okay, along with the pheromones, you are going to see visual attractants and olfactory attractants. Then here, the name itself is indicating the first one is food-based attractants. These are like sugars, proteins, and specific food items that an organism finds appealing. And the second one is pheromones. These are the chemical signals released by the organisms in order to attract the same species for mating. And the third one is visual attractants. These are the colors or the patterns that attract organisms. Examples, flowers attracting pollinators. Okay. And the fourth one is olfactory attractants, scents that lure organisms. Examples, second class fragrance attracting bees. These are the types of the attractants. And what are the uses of attractants? These attractants are used in agriculture and pest control, usually in research and fishing even. The first one is agriculture. How these attractants are used in agriculture? They are used to lure pests into crops, reducing their population and protecting crops. And the second one is pest control. These attractants help in monitoring and managing pest populations in homes and gardens. And the third one is research. These attractants used in research in order to study the behavior and ecology of various organisms. And the fourth and the last one is fishing. Baits often contain attractants to lure fish. All these are the uses of attractants. The first one under the attractants, what we have studied is types of introduction about the attractants, what are attractants, and after we have studied about types of attractants and the uses of attractants. And what are the examples of the attractants? Already we have studied about different types of the attractants. Here specifically examples were mentioned. There are insect pheromones which are used in traps to monitor and to control pest population. And the second one is, you no need to write all these examples, you write any two examples in your exam. But if you could teach more than two examples, at least you will write two examples in your exam. So make sure that you are remembering at least two examples in all the topics to highlight its importance. And coming to the second one, fish bait. These are the substances like blood or chum used to attract fish. And the third one is flower scents used to attract pollinators like bees and butterflies. And the fourth one is methyl-eugenol, a natural compound found in various plants, primarily used as an attractant for fruit flies, especially the oriental fruit fly. And fifth one is ethyl 3-isobutyl 2,2-dimethyl cyclopropane carboxylate, which is a synthetic compound used as an attractant for certain pest species, particularly in agricultural pest management. Example, coconut rhinoceros beetle. And the last one is dimethyl dioxospira undetained. It is used as an attractant for the yellow fever mosquito, that is Aedes mosquito, which causes dengue fever. This is about the example. These all are the examples of attractants. And coming to the environmental considerations, these attractants may lure unintended species, potentially disrupting ecosystems. From the beginning, we are talking about these things because when we are want to target some of the species, without our notice, we also may target the non-target species also. Because of that, there will be a disturbance in the ecosystem. So while we are using these things, while we are using the pesticides, while we are using any insect control agents, we have to take the precautions to protect the non-target species. Okay, so here attractants may lure unintended species, potentially disrupting ecosystems. And some synthetic attractants can leave residues that impact the environment. So we must be careful when we are using this. And coming to the regulations and safety, many attractants, especially those used in agriculture and pest control, are regulated to ensure they are safe for use and do not harm non-target organisms or the environment. This is all about the attractants. Here we have covered about how these attractants are playing a major role in control, how these attractants are using as insect control agents, and what are the types, what are the examples, how to be used, in what level, how to be used in order to control the pests. And the fourth, and the next one is sex pheromones. See, we all know about the sex pheromones. Just now, under the types of attractants, we have studied about the pheromones. Okay, here these pheromones are powerful tools in the management and study of various species, and the specificity and the effectiveness in attracting individuals make them invaluable in agriculture, research, and conservation. And so, by harnessing these natural pheromones, we can develop sustainable and targeted approaches to pest control, wildlife management, and to understand animal behavior. This is the importance of pest control. If you could see here, the mode of action. We all know that these sex hormone pheromones are chemical signals which are secreted by an organism in order to attract individuals of the opposite sex, facilitating mating. And these pheromones play a crucial role in reproductive behaviors across a wide range of species, including insects, mammals, and marine organisms. And they are highly species specific, allowing for precise communication and attraction within the same species. Coming to the mode of action of the sex pheromones, here it is given in the form of a steps. First, the sex pheromones will be produced, and then after it will be released. Then after you can see the dispersal of these sex pheromones, then identification or detection, finally the behavioral response. All these are the steps which you are going to see in the sex pheromones mode of action. What happens in the production and releasing of these sex pheromones? These are produced by special glands and released into the environment. In insects, for example, these glands are often located on the abdomen region. Okay, once released, once the sex pheromones are produced and released, these will be dispersed through the air or water depending on the species. The concentration and dispersal pattern can vary based on environmental factors like wind, humidity, and temperature. And once it gets dispersed, the target organism detects the pheromone through specialized sensory organs. In insects, this is typically done by antennae, which are equipped with olfactory receptors which sense smell, which are sensitive to specific pheromone molecules. Once the insect recognizes upon recognition, the organism exhibits a range of behaviors aimed at locating the source of the pheromone. This might include flying, walking, or swimming towards the source. The response is often innate and hardwired into the organism's neural circuitry. This is all about the mode of action of sex pheromones, how it will be produced and released, how the target organism recognizes this, and how it responds towards that particular chemical substance, which is called as a pheromone. Here I have given you some of the examples of the sex pheromones. The first one is agricultural pest management, and the second one is research and behavioral studies, and the third one is conservation and wildlife management. I already told you before I start with the sex pheromones. These pheromones specificity and effectiveness in attracting individuals make them invaluable in agriculture, research, and conservation. So, by harnessing these natural signals, we can develop sustainable and targeted approaches to pest control, wildlife management, and to understand animal behavior. So, here, uh, uh, as we have studied this, the importance in pest control, wildlife management, and etc. Under that, we are taking few examples in order to understand these concepts very clearly. And the first one under the agricultural pest management is crawling moth. Cydia pomonella. It is a sex pheromone. The sex pheromone of the crawling moth is used to monitor and control populations in apelopters. These synthetic pheromones are placed in traps to lure male moths, reducing mating success and subsequently lowering the pest population. This technique is known as mating disruption. Okay, but when you are writing the same in your exam, try to highlight some of the important points like mating disruptions, mating mass trapping, all these things. Okay, here we have taken some examples in order to study about this. Okay, what are the benefits of this sex pheromone of the crawling moth? It reduces the need for chemical insecticides, making pest control more environmentally friendly and sustainable. And the second example here is oriental fruit moth, Rhaphrodita molesta. The pheromone from this particular moth, oriental fruit moth, traps are used to monitor and disrupt the mating patterns of the oriental fruit moth in it dispenses emitting the female sex pheromone, which are placed throughout the object to confuse males, preventing them from finding females. And this helps in reducing crop damage and controlling pest populations without relying heavily on chemical pesticides. Okay, this is all about the agricultural aspect. And the second one is research and behavioral studies, where the example here given as European corn borer, Australia luvialis. The researchers used the sex pheromone of the European corn borer to study its mating behavior and population dynamics. The pheromone-baited traps are deployed to capture males, allowing scientists to monitor population levels and study migration patterns. It provides valuable data for developing more effective pest management strategies and understanding pest biology. This is about research and behavioral studies. And the third one is conservation and wildlife management. Under that, the example is elephant. Sex pheromones are used in conservation efforts to manage elephant populations and reduce human-elephant conflicts. Pheromones can be used to lure elephants away from human settlements or to specific areas where they can be monitored or managed. And coming to the applications, it helps in protecting both elephant populations and human communities, promoting coexistence. Okay, so here we have studied about three different aspects: research-oriented aspect, agricultural aspect, and the last one is conservation and wildlife management. So here insect repellents are the chemical substances which are applied to stain, clothing, and other surfaces to deter insects from landing or biting. They are widely used to prevent insect-borne diseases and discomfort caused by insect bites. Repellents are crucial in areas where insects are vectors for diseases such as malaria, dengue, etc. Coming to its mode of action, coming to the mode of action of sex pheromones, olfactory interference, second one is sensory disruption, and the third one is behavioral modification. Coming to the olfactory interference, most insect repellents work by interfering with the insects, that is, sense of smell, masking the attractive odors emitted by humans or animals. Second one is sensory disruption. Repellents can also act on the insect sensory receptors, making it difficult for them to detect potential hosts. And the third one is behavioral modification. Some repellents cause insects to exhibit avoidance behavior, moving away from the treated area or host. All these are the mode of action. Under sex pheromones, we have studied about what are the sex pheromones and the examples of sex pheromones in the area of agriculture, in the area of research, and in the area of wildlife management. Coming to the mode of action, we have studied about three different things: olfactory interference, second one is sensory disruption, and the third one is behavioral modifications. And here are the examples of the forms of insect repellents. Lotions and creams which are directly applied to the skin, okay, sprays and aerosols used on stain, clothing, and surrounding areas. There are wipes here, pre-soaked wipes for easy application to skin. Then wearables, example bracelets, patches, or stitches impregnated with repellent chemicals. And the last one is standees and coils, burned to release repellent smoke or vapor in the environment in order to control the insect population. And here there are some of the examples of the chemicals which are acting as the repellents. The first one is DEET, and the second one is Stevia 3023, and the third one is IR 3535, and the fourth one is oil of lemon eucalyptus, and the fifth one is permethrin. Okay, all these are the examples of the different types of chemicals which are acting as a repellents. Okay, but in your syllabus we have only two different types of the chemicals as an examples. But as a complete information, just I want to give you the high note on these chemicals as repellents. The first one is DEET, which is a broad spectrum repellent effective against mosquitoes, ticks, fleas, etc. Generally, they are safe when used as directed, but high concentrations may cause skin irritation, and rarely it shows neurotoxic effects. And the second one is petardin. It is effective against a wide range of insects similar to DEET, but with a more pleasant smell and less greasy feel. It shows low toxicity, and minimal skin irritation was reported. The third one is IR 3535, which is also called as ethyl butyl acetyl amino propionate, which is effective against mosquitoes, ticks, and flies. It shows low toxicity and generally well tolerated. And the fourth one is oil of lemon eucalyptus, which is a natural repellent effective against mosquitoes and ticks, generally safe with low toxicity, skin irritation possible in sensitive individuals. And the fifth one is permethrin. It is used to treat clothing, hair, and bed nets, which is effective against mosquitoes, ticks, and many arthropods, that is the insects. It shows low toxicity to humans but highly toxic to cats and aquatic life. Okay, these all are the examples or the chemicals which are acting as a repellents. Here I have given you acute toxicity with these examples. You just go through it after I send you the notes. Okay, this is all about your sex pheromones. In sex pheromones, we have studied about different things, and moreover, we have studied about the chemicals which are acting as a repellents also. Chemicals which are acting as chemicals which are acting as a repellents. Under this, we have studied different aspects, different chemicals. Example, DEET, and we have even studied about petardin and eucalyptus oil, etc. All these things we have studied. Coming to Not only that, these insect repellents were a crucial component of World War II. I think many soldiers used these insect repellents in order to protect them from different types of the insects. And the development and use of these repellents not only had a significant impact on different military operations, but also laid the foundation for post-war advancements in public health. This is all about the insect repellents, and moreover, whatever the things which we have studied, the mainly DEET, the chemical which is acting as a repellent, which the DEET. Uh, After the World War II, post-war, DEET developed during the conflict became widely used to its efficacy and long-lasting protection. And these repellents are essential for maintaining troop health and operational efficiency in tropical environments. And so the advancements in repellent technology during the war laid the what there's a groundwork for future innovations which are significantly benefiting for both military as well as the civilian populations in different types of disease preventions which are caused by different types of the insects. So this is about the insect repellents. So by this we would able to understand that these are the insect repellents which are essential for preventing insect bites and reducing the risk of insect-borne diseases. Okay, this is all about your um, insect repellents. The first one we have studied about attractants, and the second one is sex pheromones, and the third one is insect repellents. And the last one today we are going to talk about, I mean, under the synthetic insect control agents, we are going to talk about the fourth one, antiphedants. What are antiphedants? These are the substances which deter herbivores, particularly insects, from feeding on plants. These chemicals are either naturally produced by plants as a defense mechanism or synthetically manufactured for use in agriculture and pest management. And these antiphedants do not kill the pests but instead make the plants unpalatable, thus protecting crops from damage. Now, coming to the mode of action of these antiphedants. First one is taste modification, and the second one is olfactory disruption, and the third one is physiological effects. What are the what are the different types of antiphedants which we are going to use? The first one is azadirachtin, and the second one is rotenone, and the third one is pyrethrin. All these things we have studied previously in our previous classes when we are talking about the different types of the chemicals which are used as a pesticides. Here, both the chemical pesticides as well as the natural pesticides are included. Coming to the mode of action, the first one is taste modification, where antiphedants alter the taste of the plant, making it unpalatable or unpleasant for the insect to feed. When an insect takes a bite, the taste receptors which are present on the insect's mouth parts detect the antiphedant, leading to an aversive response. Second one here is olfactory disruption. These antiphedants emit odors that repel insects, preventing them from approaching the plant. And these odors can interfere with the insect's ability to locate or recognize the plant as a food source. And the third one here is physiological effects. There are certain antiphedants which disrupt the digestive or metabolic processes of the insect, making it difficult for them to derive nutrients from the plant. This can lead to reduced growth rates and reproductive success in the pest control. This is all about the antiphedants. And examples of the antiphedants, the first one is azadirachtin, which is derived from neem plant. This disrupts feeding and growth by interfering with the hormonal systems of insects. It is widely used in organic farming as a natural insect repellent and growth regulator. And the second one here is rotenone, which is extracted from the roots of certain tropical plants such as Deris and Lontrocarpus species. And this acts as a feeding deterrent and also has insecticidal properties by disrupting cellular respiration, which is used in organic farming but with caution due to its toxicity to aquatic life. The third one is pyrethrin. These are extracted from the flowers of chrysanthemum plant. These repels insects by affecting the nervous system, causing paralysis. And they are commonly used in household insecticides and in agriculture as a natural pesticide. And the fourth one is capsaicin, which is an active component of chili peppers, which causes irritation and a burning sensation in the mouth parts of insects, deterrent feeding, which is used in animal repellents and some pest control products. And the last one examples of antiphedants are ultralights. These are nicotine is an example under the ultralight. This is a naturally occurring ultralight uh, obtained from the tobacco plant. It affects the nervous system of the insects, leading to feeding deterrence and eventual death at higher concentrations. Historically, it is used in pest control, though its use has declined due to its toxicity to humans and beneficial insects. So this is all about the antiphedants. Under antiphedants, we have studied about what are antiphedants, what is its mode of action, what are the different types of the antiphedants are used in agriculture in the pest control management. So by this we could able to understand that these antiphedants are major type of insect control agents which plays a crucial role in integrated pest management and offering a way to protect crops from insect damage without relying solely on lethal insecticides and by deterring the feeding, these antiphedants help maintain crop health and yield while minimizing environmental impact and promoting sustainable agricultural practices. This is all about antiphedants. So, by this, we have completed the sympathetic insect control agents. Under the sympathetic control agents, we have studied tumor stimulants, under that alkylating and non-alkylating agents, and then um, insect insect growth regulators and juvenile hormone levels and endocannabinoids, attractants, sex pheromones, and insect repellents. Finally, antiphedants. This is all about the synthetic insect control agents. By this, we have completed that unit, and today we are going to start with the complete information about the next important chapter. Already on this, we have covered the overview of it. That is metabolism, resistance, and residues of pesticides. Did you all understand this topic, what we discussed in our previous classes about the synthetic insect control agents?
Yes, ma'am. Yes, ma'am.
Okay. And apart from that, in our previous class, we even have covered the overview of metabolism, resistance, and residues of pesticides, where almost 5 different subtopics are given. We even covered about those 5 different subtopics. The first one is photochemistry of pesticides and insect resistance in insecticides, etc. Okay, and today we are going to talk in detail about the photochemistry of pesticides. And now I'll try to open my screen and close the screen. One minute. Can you see the screen? Hello?
No, ma'am.
No, ma'am.
No. Okay, 1 minute, Amma. Mm, 1 minute, please. Yes. Amma, can you see the screen now?
Yes, ma'am. Yes, yes, madam.
In our last class, we have covered about the photochemistry of pesticides, where I have given you the overview of the photochemistry of pesticides. What is the mechanism of photodegradation? By using different examples, we try to understand what is the photochemistry of pesticides. Can you all see the examples? What I am scrolling here? We have covered all these things in our previous class. But today we are going to directly enter into the topic. The first one is basic process in photochemistry. Is that okay, or should I give you a brief overview of it? See, in our last class, we have studied, I have given you the overview of metabolism, resistance, and residues of pesticides. As we all know that these are interrelated aspects which significantly influence their efficacy, environmental impact, and safety. So, by understanding the processes like metabolism, resistance, and residues, uh, we can develop effective pest management strategies. Not only that, can ensure food safety and protecting the environment. This is we have studied, this is what we have studied under metabolism, resistance, and residues. And coming to the photochemistry of pesticides, the first one under this, we have completed few important things under the photochemical reactions. Though it is a photochemistry of pesticides, we tried to cover few of the important things as an introduction in our last class, where we have studied about how the phytolysis occurs, how the reaction takes place using the energy as a sun, using the energy in the form of using sunlight as an energy source, where we have studied about different types of the reactions like Norrish type 1 and type 2 reactions. And we even studied about the dehydrohalogenation and isomerization reactions. How these reactions are playing a major important role in environmental degradation and transformation of pesticides. All these things we have studied in our previous class. Not only that, we all know that with our previous knowledge, these reactions helps to break down the complex pesticides molecules into very simple molecules, which are very often less toxic, and thereby influencing their persistence and impact on the environment. So this is all we have studied in our previous class, and today we are going to talk specifically in detail part of photochemistry of pesticides. Under that, in order to understand how these things occur with different types of the pesticides, today we are going to cover the basic processes in photochemistry. What are the basic processes in photochemistry? How the light will be absorbed, how the how the electrons reaches to the excitation state, and what are the factors which affect the pesticides during further decomposition studies. All these things we are going to study today. Under that, the first one is basic process in photochemistry. We know that photochemical reactions of pesticides are chemical processes which involves in different factors. The first one is primary process, secondary process, and the absorption of light will be quantified in the form of a quantum. This is a unit of energy. All these things we are going to study in this particular unit. This is very simple. If you could able to recollect the things what you have studied in your previous classes, basic, if you could have the basic knowledge about the photosynthesis and all, at least you could be able to connect to these things what we are going to study today. Okay, and coming to the basic process in photochemistry, this plays a major important role in degradation and behavior of pesticides. Our concentration is to deal with pesticides or insecticides. Okay, but here it plays a major important role. How this photochemistry plays a important role in the degradation and behavior of pesticides in the environment. When pesticides absorb sunlight, that is the UV rays in a form, I mean, as an energy source, they undergo various photochemical processes that can lead to their transformation, degradation, and detoxification. Because when we are using the pesticides in order to control the pests, that may damage the things which are the leftover pesticides or the leftover insecticides that remain in the some of the parts may remain in the soil or in the plant parts that may cause harm to the environment. So not only we should study about the importance of pesticides, how to use the pesticides to control the pests and all. Along with that, we should also study how to degrade the pesticides, how to detoxify the pesticides to protect our environment. All these things we are going to study under this unit, that is photochemistry of pesticides. Under that, the first one is the primary process. What is the primary process? Under the degradation, I mean, in this particular process, transformation, degradation, and detoxification, in this particular process, the first step is primary process, where you are going to see the absorption of light. How exactly it will be happen? The pesticides which are present in the environment or which are present in the plants, whatever we have sprayed to control the pests, absorbs photons from sunlight. Okay, you know what are photons? Pesticides absorb photons from sunlight, promoting electrons from the ground state to an excited state. And the energy of the absorbed photon must match the energy difference between the ground state and the excited state of the pesticide molecule. There are some examples which are given here. The first one is chlorpyrifos, which is an organophosphate pesticide, which we have studied already in our previous classes. Okay, when we are studying about the molecules with different chemistry, we have studied about different types of the molecules like neonicotinoids, meristoxin, spinosad, averimetin, chlorphenapir, indexotarb, pymecrozine, etc. Okay, so not only these things, we even studied about the other types of the ultralights also. But here, as an example, there are many examples, but just as a one example, here we have given chlorpyrifos, which is an organophosphate pesticide that absorbs the sunlight that leads to the photo degradation after it gets exposed to the UV light, which is from the sunlight. And once the absorption of the light was observed with respect to the pesticides, the electrons from the ground state reaches to the excited state. What is excited state formation? Upon absorbing light, pesticides transition to excited states, which can be singlet or triplet states. What is a singlet state? The excited electron maintains its spin paired with the ground state electron, and the triplet state, the excited electron has parallel spin to the ground state electron. We studied all these things about in our lower classes when we are talking about the number of electrons, how these electrons move around the nucleus, how the electrons will be filled in an orbital, all these things. Okay. The next one is emission and absorption process. What is an emission and absorption process? Here, the light or the radiation in a vacuum travels at different velocities. We all know about this, but depends upon the type of the pesticide, it varies. Okay, so coming to the absorption, these pesticides absorb light at specific wavelengths. As I told you, different pesticides has different capacities to absorb the light. The light has different wavelengths. We already we have an idea about the electromagnetic radiation, where different lights are there, visible light, UV rays, or UV light, all these things, where the wavelength ranges differently. Okay, here the pesticides absorb light at specific wavelength, which can be studied through absorption spectra. Example, pyrethroids, which is a class of synthetic insecticides, have a specific absorption bands in the UV region, which ranges from 260, which is at 260 or 280, something like that. Okay, and the second one here is fluorescence. Some pesticides may return to the ground state by emitting photon, a process known as fluorescence. Fluorescence occurs quickly and usually at longer wavelength than the absorbed light due to energy loss. Example, fluorescent, which is a fluorescent marker used in pesticide formulations in order to track their presence and the degradation of the pesticide. And the third one is fluorescence or phosphorescence. What is phosphorescence? Emission of light from the triplet state to the ground state, occurring more slowly than fluorescence. Phosphorescence is less common in pesticides but can be observed in specific cases. This is all about emission and absorption process. Here, under the emission and absorption process, we have studied three different things. The first one is absorption, and the second one is fluorescence, and the third one is phosphorescence, where you have seen the emission of light or the absorption of light. Finally, our aim is to detoxify the pesticides. And the next one here is excited state process. What is an excited state process? Here you can see different types of this things where excitation, sensitization, all these occurs under the excitation state process, where the first one is non-radiative decay. This non-radiative process is a internal is of two types, internal conversion, and the second one is inter-system crossing, where non-radiative process shows where an excited pesticide molecule transitions between electronic states of the same spin multiplicity, losing energy through vibrational relaxation. I'll show you these things with some examples and videos. I'll try to share the links of the videos, how to understand these processes, basic part of photochemistry. Because all these things we have studied, we have the knowledge, but if you're able to watch those videos, it will give you the clear picture how exactly the light gets absorbed by different types of the pesticides, what happens exactly in the pesticides, what are the different types of the chemicals that can be activated, okay, how the shift can occur between the states, that is the initial state, ground state to the excitation state, when it gets activated, and how the detoxification occurs, and all these things. Detoxification means it will become inefficient. It does not cause any harm. Okay, all these things we are going to. It would be easy for you to understand after I share the videos regarding this. Okay, now the second one is radiative decay. Already we have studied about fluorescence and phosphorescence here under the emission and absorption process. Here also under excitation state process, the same examples were given under the radiative decay. How the non-radiative decay occurs, how the radiative decay occurs. Non-radiative decay process, where you have seen two things, internal conversion and inter-system crossing. In the internal conversion, excited pesticide molecule transitions between electronic states of the same spin multiplicity, losing energy through vibrational relaxation. But in the system crossing, where an excited pesticide molecule transitions between states of different spin multiplicity, often lead to phosphorescence. So these are the two different types which are going to see non-radiative decay, and the second one is radiative decay, where fluorescence and phosphorescence will be seen. And the third one is energy transfer, which under that, if you could see first example, FRET, Foster resonance energy transfer, which is a process where an excited pesticide molecule transfers energy non-radiatively to a nearby acceptor molecule through dipole-dipole interactions. And the second one is Dexter energy transfer, which involves the exchange of electrons between donor and acceptor molecules requiring close proximity. And the fourth one is photo-induced electron transfer. The excited state pesticides can transfer an electron to or accept an electron from another molecule, leading to the formation of charged species. Example, photodegradation of the herbicide atrazine, where photo-induced electron transfer leads to the formation of reactive intermediates. And the fifth one is photochemical reactions. Under that, the first one is photodissociation, where you are going to see the absorption of light, which leads to the breaking of chemical bonds in the pesticide molecules, forming smaller molecules. Example, breaking down of DDT under UV light to form DDE and DDD, which are less toxic but more persistent. And the second one is photoisomerization. Photoisomerization means light-induced structural changes in the pesticides. There is no change in the molecular formulae, but structurally there will be a change, depends upon the environment. Example, isomerization of the herbicide metolachlor under sunlight, altering its effectiveness and degradation rate. And the third one is photocycloaddition, where light-induced reactions of two different unsaturated molecules form a cyclic product. Example, formation of cyclobutane rings in certain photodegradation pathways of pesticides. Why photodegradation has to occur? In order to reduce its toxicity levels. There are different types of the examples which are listed here. The first one is chlorpyrifos, and the second one is atrazine, and the third one is metolachlor. This chlorpyrifos absorbs UV light, leading to the formation of reactive intermediates which degrade into less toxic compounds. Involves photodissociation and photo-induced electron transfer process. And the second one is atrazine, which undergoes photodegradation through photo-induced electron transfer, forming radicals and other reactive species. This process is crucial in reducing its persistence and toxicity in the environment. And metolachlor, which experiences photoisomerization when exposed to sunlight, impacting its herbicidal activity and environmental fate. All these are the examples of the first one, that is basic how the photodegradation occurs in different types of the pesticides. These are the pesticides, how the photodegradation occurs. By this, we have completed about the basic processes in photochemistry. Under this particular thing, we have studied how the electrons gets excited in a particular compound, example, pesticides, okay? How a series of complex reaction processes occurs, which determines the stability, effectiveness, and the environmental impact. So, by understanding these primary processes, like where we have studied about emission, absorption, and excited state behaviors, all these are very much essential for developing safer and more effective pest control strategies. And not only that, it also helps in mitigating the environmental risks which are associated with the pesticide use. Our target is to reduce the effect of the pesticides which we have used for the pest control. So this is all about the photochemistry of the pesticides, what we have studied under that, the basic processes in photochemistry. Do you have any doubts here? You please ask me now. Otherwise, we'll enter into the other aspects where the factors which affects pesticide photodecomposition studies. Hope it is there. Yes. Here, this is the next topic under the photochemistry of pesticides. That is the factors which affects the pesticide photodecomposition. There are different types of the factors. See, if anything is occurring, some of the factors that affects its process. Like that here also, photodecomposition and photodegradation should occur in different types of the pesticides in order to protect the environment, because some of the pesticide remains will be seen in different types of the soils and in the plants. In order to remove all these things from it, we have to use different types of the strategies, especially we are using here the direct light, UV light or the sunlight as an energy source in order to make changes in the organism, so that the organism sorry, in order to make changes in the chemical compound so that it will not show its toxicity. This is all about your basic processes in photochemistry. Do you have any doubts? If you have any doubts, please try to clarify with me right now. Anyhow, I'm going to share you some of the video links where at least details about the photochemistry will be given. Hello, am I audible? Hello.
Yes, yes, madam, it is audible.
Am I clear? Do you have any doubts? Please feel free to clarify your doubts.
Yes, madam, it is clear.
Okay. Now the second one here is there are different types of the factors which involves with pesticide photo decomposition studies. The first one here is, yeah, what happened? Okay. Photo decomposition of pesticides refers to their breakdown or transformation induced by light exposure, which can occur in soil, water, or in plant surfaces. There are several factors that influence the photo decomposition of the pesticides, including light sources. There are different types of physical state of substrates, etc. So here the first one is there are different types of the models that can be used for studying in photo decomposition. The name itself is indicating photo decomposition, decomposition of a substance using the photon or the light. Okay, so here the first one is laboratory studies where controlled environments allow precise monitoring of pesticide degradation under specific light conditions. Example, pesticide solutions or thin films exposed to UV lamps or simulated sunlight in quartz reactors. Second one is environmental simulations, where we mimic the natural conditions which we are seeing in the environment more closely, integrating factors like temperature, humidity, and varying light intensities. Example, outdoor exposure studies using environmental chambers or field plots. Okay, just we'll try to understand how these models are using for the photo decomposition process. The first one is the laboratory studies in the laboratory, and the second one is using environmental chambers or field plots. And the second one is the light source. How the light sources are affecting the pesticide photo decomposition. The first one is UV lamps. What are UV lamps? It will emit UV radiation. There are different types of UV radiation. UVC also. Okay, whose primarily UVA, whose wavelength is this? Ranges from 320 to 400, UVB 280 to 320 nanometers wavelength, which simulate in the sunlight. Example, mercury vapor lamps used in controlled laboratory experiments. And the second one is simulated solar radiation. Full spectrum light sources that replicate sunlight, including visible and UV wavelength. Example, xenon arc lamps, which are used for environmental simulation studies. All these are about the light sources. Two things we have studied: UV lamps, which are available in the market, and the second one is simulated solar radiation. How these are showing its effect on pesticide photo decomposition. Okay, and the third one is physical state of substrate. The first example is soil, and the second example is water. Pesticides can absorb to soil particles, influencing their availability for photodegradation. Example, absorption of atrazine to clay minerals affecting its photochemical fate. Second one is water. Dissolved or suspended pesticides undergo direct photodegradation depending on water depth and clarity. Example, photolysis of herbicides in surface waters affecting aquatic ecosystems. And the fourth one here is after the physical state of substrate. First one we have studied about the models. Second one we have studied about the light sources. And the third one is physical state of substrate. And the fourth one is sensitization or sensitizers. There are two examples which we have given here. The first one is natural organic matter. This absorbs sunlight and transfer energy to pesticides, enhancing their photodegradation. Example, humid substances in soil or water are sensitizers for pesticide breakdown. And the second one is metal ions. These metal ions catalyze photochemical reactions or generate reactive oxygen species under light exposure. Example, iron ions in soil catalyzing the photodegradation of pesticides. What are these reactive oxygen species? During less oxygen, the diatomic oxygen gets converted into oxygen atom and it will react with, as it is unstable, it reacts with any of the molecule in a cell and it will tries to affect the cellular activities and eventually it leads to the death of the cell. So all these are the examples. All these two are the examples of sensitizers. And fifth one here is photooxidation. How this photooxidation affects the decomposition or degradation of the pesticides. The first one is direct photolysis. What happens in the direct photolysis? Direct absorption of light by pesticides leading to bond cleavage or structural changes. Example, photodegradation of atrazine involves direct cleavage of chloroethylamino group, which forms hydroxylated products. And the second one is photoinduced electron transfer. Electron transfer from the pesticide to a nearby molecule or surface generating radicals. Example, PET-mediated degradation, which is photo-induced electron transfer degradation of organophosphate pesticides like chlorpyrifos forming reactive intermediates. And the third one here under photooxidation reactions are photooxidation by reactive oxygen species, generation of ROS, that is a singlet oxygen, hydroxyl radicals in the presence of light which oxidizes pesticides. Example, photodegradation of glyphosate involving hydroxyl radical attack on the phosphonomethyl group, which leads to the breakdown of the pesticides. And the fourth one is photocatalysis, where we are going to use the semiconductor materials, that is titanium dioxide, that generates reactive oxygen species under light, accelerating pesticide degradation. Here there is an example, titanium dioxide, which is a photocatalytic compound. This performs photocatalytic degradation of different types of neonicotinoids, which enhances the breakdown in water treatment process. This is all about different types of the factors which are affecting the pesticides photo decomposition or the photo degradation. Under this, we have studied about two examples under each factor. The first factor is model, different types of the models like in labs, all these things. And the second one is light source, UV lamp, all these things. And the third one is physical state of the substrate. And the fourth one is sensitizers. And the fifth one is photooxidation. So these are very much essential for assessing the environmental fate and developing mitigation strategies. So from laboratory models and light sources to the physical state of substrates, sensitizers, and photooxidation reactions, each factor plays a very major important role in determining how pesticides gets break down under light exposure. This is all about the factors that are affecting the pesticides photodegradation. And the next topic is chlorinated. I don't know, 1 minute. Yes, chlorinated hydrocarbon pesticides. Okay. Under the chlorinated hydrocarbon pesticides, we're going to talk about three different things. Okay. The first one is DDT and envelopes, and the second one is HCH, and the third one is cyclodine. Okay, these three things we are going to study under chlorinated hydrocarbon pesticides. What are chlorinated hydrocarbon pesticides? We already have an idea about these chlorinated hydrocarbon pesticides from our previous classes where we have studied about different types of the sprays or the powders which we have used in order to control the pests. So here examples are DDT and HCH and cyclodine pesticides, which involves their behavior under light exposure, which can lead to degradation, transformation, or the formation of toxic intermediates. The first one is DDT. We all know about the DDT. It is made up of benzene rings with chlorine and hydrogens are attached. There are different types of the examples which we are seeing here, DDT and DDE, which are breakdown products of DDT. Okay, how this DDT break or undergoes cleavage to form DDD and DDE. Here you can see direct photolysis. DDT absorbs UV light, primarily UV and UVC, leading to direct photolysis. UV radiation breaks carbon-chlorine bonds which are present in the DDT, forming DDE and DDD as a major breakdown products. And here, from the product, DDE is persistent, bioaccumulates in food chain, and poses risk to wildlife and humans. And the second example here is HCH. Okay, HCH refers to a group of isomers, that is hexachlorocyclohexane, including alpha HCH, beta, gamma, and delta. Here there is an example which is given, that is gamma HCH, which is also called as lindane, which is the most common and toxic isomer. This lindane undergoes photochemical isomerization under UV light, converting to other HCH isomers and breakdown products. UV radiation induces changes in molecular structure, altering biological activity and toxicity. And isomerization and degradation reduce this gamma HCH lindane persistence, but may still produce toxic intermediates. And the third one under chlorinated hydrocarbon pesticides include cyclodine pesticides. Okay, examples are aldrin and dieldrin. These are the cyclic compounds with multiple fluorines, which are known for their persistence and toxicity. So the aldrin converts to dieldrin through photochemical reactions. First, the aldrin absorbs UV light, leading to photoisomerization to dieldrin. UV radiation induces bond cleavage and rearrangement of the chlorines which are present in the aldrin, forming dieldrin, which is a major breakdown product. This dieldrin is highly persistent, bioaccumulative, and toxic, posing risk to ecosystems and human health. There are some examples of the photodegradation reactions. Already what we have studied in our just previous slide are given in a short form for easy remembrance. That's it. This is all about the chlorinated hydrocarbon pesticides. Okay. here we know that by this we could able to know that it's a complex process of photolysis where the UV gets absorbed by different types of fluorinated hydrocarbon pesticides, and you are seeing photolysis and the isomerization, etc. When the pesticide gets exposed to the UV light, and this leads to the breakdown of the pesticides that vary in toxicity and persistence, which are influencing environmental fate and risk to the ecosystem. This is all about chlorinated hydrocarbon pesticides. And the next one here is organophosphate pesticides. What are organophosphate pesticides? Already we had an idea about what are organophosphates. These things we have studied in our previous slides. These are the pesticides which are used to control the pests. All these things are acting on the nervous system of the insects in order to kill them, in order to demolish, in order to remove them. Okay, coming to the organophosphate pesticides, the photochemistry of organophosphate pesticides, specifically phosphorothioates and phosphorodithioates, involves their behavior under light exposure, which can lead to degradation or transformation. There are two examples which we have just listed here. The first one is phosphorothioates. They have central phosphorus atom bonded to 3 oxygen atoms and 1 sulfur atom. Examples are parathion, malathion, and femthion. Parathion is used as an insecticide and acaricide. Malathion it's a broad spectrum of insecticide and mosquito larvicide. Femthion is an insecticide used against agricultural pests. How the photochemistry occurs with these organophosphate pesticides? First, absorption of UV light by phosphorothioates can lead to direct photolysis, where UV radiation can break phosphorus and sulfur bond, resulting in the formation of breakdown products such as phosphoric acid and sulfur dioxide. And this photodegradation reduces the concentration of active pesticide molecules in the environment over time, and some breakdown products may still retain toxicity, posing risk to non-toxic organisms. But somehow we could be able to control its effect in the environment by photolysis. And the second example under the organophosphate pesticide is phosphorodithioate pesticide. It also has a phosphorus atom in the center bonded to 2 sulfur atoms and 2 oxygen atoms. Slightly, there is a difference between phosphorothioates and phosphorodithioates. Examples of the phosphorodithioate pesticides include disulfotan and phorate. Disulfotan is used as an insecticide and acaricide. Phorate is a soil insecticide used against a wide range of pests. What is the mechanism of the UV light on phosphorodithioate pesticides? Similar to phosphorodithioates can undergo direct photolysis under UV light, where you are going to see the breaking of phosphorus and the sulfur bonds, leading to the formation of sulfur dioxide and other breakdown products. Coming to the environmental fate, while phosphorodithioates can degrade under light exposure, some breakdown products may persist in the environment. Monitoring and regulation are crucial due to potential environment persistence and toxicity of degradation products. There are some examples which we already studied here. As a glance, I have given parathion, malathion, and disulfotan. All these are the examples of organophosphate pesticides, where we have learned about phosphorothioates and phosphorodithioates, which involves their transformation under the light exposure, which leads to the formation of less toxic or inert breakdown products. So by this, we can able to assess the environmental risks, we can manage the pesticide applications, and we can develop the strategies to mitigate their impact on the ecosystem as well as on the human health. So this is all about organophosphate pesticides. And the next one here is carbamate. Okay, we have studied about even carbamates in our previous classes. We know how to use the carbamates. We know the effect of mode of action of carbamates. But today we are going to talk about the photochemistry of these pesticides. Here we are going to talk about the carbamate. These are the pesticides which are a class of insecticides and fungicides that contain a carbamate functional group. They are widely used in agriculture due to their effectiveness against broad spectrum of pests and relatively low toxicity to mammals compared to other pesticide class. If you could see the chemical structure, it have a carbamate functional group attached to various aromatic and aliphatic structures. Examples are carbaryl and methionine. Carbaryl is a broad spectrum insecticide used against pests in agriculture, forestry, and public health. Methomyl is a systemic insecticide used in agriculture to control chewing and sucking insects. If you could see its mode of action with respect to photochemistry, carbamate pesticides absorb UV light, primarily in the UVA and UVB ranges, leading to direct photolysis. UV radiation can break down within the carbamate structure or induce photoisomerization. Coming to the environmental impact, photodegradation reduces the concentration of active pesticide molecules of carbamates in the environment, affecting their persistence and toxicity. That's how we could be able to protect our ecosystem. This is all about carbamate pesticides. Is that clear to you all? Here, the carbamate pesticides involves the absorption of UV light that leads to the degradation of the active pesticides molecules which are remain in the soil or in the plants, and it will break into simpler compounds so that it will be less toxic to the ecosystem. Do you have any doubts still here? Hello? Hello? Am I audible? Are you listening?
Yes, madam.
Did you all understand? Carbamate pesticides?
Yes, ma'am.
Okay. Please respond, otherwise I may think that I lost my connection. And the next one is pyrethroids. We already have an idea about what are pyrethroids. These are synthetic chemicals which are derived from natural pyrethrins, which are extracted from chrysanthemum flowers. They are widely used as insecticides due to their effectiveness against variety of pests and their relatively low toxicity to mammals. If you could see the structure, already we have an idea about the structure of pyrethroids. It has two benzene rings which are connected by a cyclopropane or cyclopropane like ring with a carboxylate ester group. Examples of pyrethroids include permethrin and cypermethrin. Permethrin is used in agriculture, public health, and residential settings to control insects like mosquitoes and ticks. Coming to the cypermethrin, which is a broad spectrum of insecticide used in agriculture and veterinary applications. And how the photodegradation occurs with pyrethroids. First, the pyrethroids absorb UV light, particularly UVB and UVC, leading to photolytic degradation. When they absorb the UV light, the UV light breaks the ester bond or other susceptible bonds present in the pyrethroids, resulting in the formation of breakdown products. Coming to the environmental impact, photodegradation reduces the persistence of pyrethroids in the environment, influencing their toxicity and ecological effects for the protection of ecosystem as well as the human health. All these are.
Excuse me, madam.
Yes.
What is the full form of UVA? Ultraviolet. A means what, madam?
Actually, ultraviolet radiation means it is not the visible light radiation. Okay, where you are going to see the range between the range at 260 to 280 depends upon the wavelength. which you are going to see in the ultraviolet radiation. It got divided into three types: UVA and UVB and UVC. Okay?
Okay, ma'am. Okay, ma'am. Thank
Already I have explained previously. UVA is ranging from 315 to 400 nanometers. UVB is ranging from 280 to 315 nanometers. And UVC is ranging between 100 to 280 nanometers. Okay? We have the electromagnetic radiation, right? Hope you have an idea about electromagnetic radiation,
you. Yes, madam. Yes, ma'am.
where you're going to see different types of the rays along with its wavelength.
Yes,
Okay?
madam.
Like how the visible range is 400 to 700 nanometers. In the same way, here also UV radiation has a range, okay, where depends upon the wavelength, it got divided into three types. Clear?
Yes, ma'am. Clear.
Okay. Now, this is all about pyrethroids. How the pyrethroids gets photonized or photodegraded using the UV light. And the last one here is rotenone. This also we have studied in our previous class when we are talking about the insecticides and pesticides, which is a naturally occurring compound which is derived from the roots and stems of plants like Deris, Lontocarpus, and Taphausia. It has been traditionally used as an insecticide and pesticide, and more recently as a botanical pesticide in organic farming. If you could see the structure of rotenone, which is a lipophilic compound with a complex structure containing several rings and hydroxylid groups. And it is derived from roots and stems of plants like Deris, Ellipta, and Lontocarpus species. Historically, it is used in agriculture to control insects and pests, and in fisheries to eliminate unwanted fish species. What is the photochemistry for this rotenone? How this can be decreased, how the toxicity of this rotenone can be decreased to save the ecosystem. Here, the rotenone undergoes photodegradation upon exposure to sunlight, primarily through direct photolysis. When the UV radiation falls on the rotenone, it breaks the chemical bonds which are present in the rotenone, leading to the formation of simpler, less toxic breakdown products. Coming to the environmental impact, the photodegradation of the rotenone reduces the persistence in the environment, affecting its bioavailability and toxicity to non-target organisms. This is all about here rotenone. Here is the detailed explanation about the mechanism, which type of UV light gets absorbed by the rotenone, how the cleavage occurs in the rotenone, what bonds will be break down in order to make the simpler compounds in order to protect the environment. This is all about rotenone. Did you all understand what is rotenone? How the photodegradation occurs and all? Here, the photochemistry of rotenone involves the degradation under UV light, which reduces its persistence and influencing its toxicity profile in the environment. Okay? Clear?
Okay, ma'am.
I guess we have completed the photochemistry of the pesticides, where we have studied about different things. Like the first one is basic processes in photochemistry, how the photochemistry works, what is the principle, how the electrons reaches to the excited state, what happens when it reaches to the excited state, what are different types of the things which we have which we see with photochemistry, like phosphorescence, fluorescence, all these things. We have studied about different aspects, emissions, absorptions, excited state process, all these things in the primary process, and what are the factors which affects the photo decomposition. Then after, we directly entered into the different types of the pesticides, how the photodegradation occurs with different types of the pesticides. Example is chlorinated pesticides and organophosphate pesticides, and we have studied about carbon-based pesticides, pyrethroids, and rotenone. All these about the photochemistry of pesticides. If you know the basic concept about the photochemistry, how it works, what all the things happens, photoisomerization, photodegradation, if you could able to understand lysis, if you could able to understand all these things with mechanism, then it will be easy for you to understand how the UV rays acts on the pesticides, how it breaks the bonds which are present in the pesticides to make into simpler compounds, and how it will be less toxic. Okay, all these things can be understand by understanding the basic principle about the photo chemistry. This is all about the first unit under the metabolism, resistance, and residues. Is it clear to you all?
Yes, madam.
Okay. Amma, I have shared the notes with your sir. I don't know whether your sir has shared the notes with you. If not, you please let me know. Was it shared with you?
No, ma'am.
No, ma'am.
Will you please ask sir once about the notes, what I have shared with him? As I don't have the access to share directly with you, and so I have shared with your sir. You please contact your sir regarding the notes, what I have shared, so that he will pass to you.
Which sir, ma'am?
Okay.
Amma?
Which sir?
Sure, sir.
Okay.
You only told me to contact Kishore sir regarding the WhatsApp group, right? I mean, you people only told me about that. So I have shared with that sir. You please contact sir for the notes. Okay. As I have completed this part, I'll try to share this notes also in 1 or 2 days, so that you will get the complete notes of the toxicology. Clear?
Yes,
Okay,
ma'am.
ma'am.
Okay. Shall we leave for the day?
Yes, ma'am.
Yes,
Okay.
ma'am.
Thank you all for your patience.
Thank you, ma'am.
Thank you, ma'am.
Good day.